So, good afternoon all. So, for the information of all, our honorable external expert for today's Ph.D. Viva Voce exam is Madam Professor Sweezee Thakur, who is Professor and Head of Department of Food Science and Technology. Honorable, she has done her BSc Home Science, MSc Food Technology, Ph.D. Food Technology from Punjab Agricultural University, Ludhiana. She remained president of the, she remains on various positions as scientist, senior scientist, and principal scientist in PAU. And she is the recipient of many honors and awards. Fellow of the National Academy of Dairy Sciences, Best Oral Presentation Awards at the International Conference in Singapore, Best Paper Award, many Best Paper Awards and Poster Awards at the International Conference in Pondicherry, Varanasi, Ludhiana, Sant Longwal Institute of Engineering and Technology. And she has been selected and participated and attended 4 international courses on food management in Israel and awarded excellent grade in those programs. She is member of the and life member of the Association of Food Scientists and Technology, Mysore, member of the Masterline Bakery Services Hindustan Lever Limited, Bangalore, life member of Indian Science Congress Association, life member of Punjab Science Congress Association. Her professional details are more than 31 years experience she is having, and she has guided more than 25 students in MSc and 16 students in PhD. And besides that, she remained minor advisor of many MSc and PhD students. She remained class in charge in PAU of BSc Agriculture and BTech Food Technology. She is teaching UG and PG courses. Besides, external examiner for thesis evaluation paper setter for the outside universities of PAU, outside PAU. She remained course coordinator for a short course, Food Laws and Standards Changing Scenario and Safety of Consumers sanctioned by ICR, which I think it was conducted in 2007. At present, she is handling 21-year training, 21 days, 3-week training program sponsored by ICR at PAU for the scientists of this food technology. And the scientists, I think, from many states are attending that conference at this training program at PAU. That is Summer Institute, yeah. I think, Summer Institute. And she is engaged in lab development, course development, teaching innovation aids, preparation of resource materials, conducted research on process product development in area of food technology, and imparted demonstration to food industry personnel in FDP programs as well as programs for farmers. She is associated with the characterization and valorization of millets and supergrains, development of technologies for convenience and nutritious food products, developed variety products and processes which have most relevance in the current scenario where diversification is pressing need. She worked in quality improvement and potential utilization of milling industry byproducts and development of variety extruded products, cold and hot extrusion having health benefits, biological activated food grains, characterization and functionality. She remained principal investigator of 2 externally funded proposals and 2 as PI, 3 and co-PI. And besides that, she is a member BSMA, Dairy Technology ICR, published more than 166 research papers, written 8 books, 1 bulletin, 5 brochures, and contributed book chapters in 31 books, review articles 44, extension articles, I think in hundreds. I think, and I think besides that, she has attended many national and international conferences. We are fortunate that we are having such a dynamic head of the Department of PAU who is with us and food technology scientist, very learned person. And I am really thankful to you on behalf of LPU and on my own behalf.
Thank you, Dr.
So, my associate dean, Madam Sweezee, very dynamic.
Yeah.
I think I rate her working as number 1 in food technology.
Exactly.
Madam Jyoti, Dr. Prince Chawla, Madam, both are great researchers, and I think this time both of them got many such these incentives of LPU. डॉक्टर साहब कितना पैसा मिला आपको इस बार इंसेंटिव का?
It's very good
incentives has been given by LPU. Our Honorable Chancellor Sir is really helping the scientists and through these incentives and encouraging the scientists to do good research.
I think they are doing good under your umbrella, na? Your guidance and your blessings. So, yeah.
Our PhD scholars are present in to see you, and under your guidance they want to learn many things. Our PhD student. So I think, madam, now I think we hand over to madam. Okay, madam, you start.
Thank you, Thank you. Is the first presentation then?
It's the first presentation.
Okay, that's fine.
Get started.
Introduce to madam.
Okay. Right. Good afternoon to all the faculty members. I would like to express my heartful gratitude towards Dr. Sweezee ma'am for taking the time to evaluate my work. And I'm also thankful to Dr. Prince Shabla, who is my supervisor, and the co-supervisor, Dr. Anil Kumar, for their support and guidance throughout my journey, PhD journey. And also thankful to all the faculty members of the LPU or the department to shaping my academic journey. And lastly, I would like to thank my juniors or my friends for always there for me. So with that, I am a PhD scholar, Sweezee Thakur, PhD scholar of Food Science Technology. Today I will present my final research viva of PhD on the topic detection of the residual packaging toxins from the food using the food industrial waste derived carbon quantum dots. So I'll give the overview of my work. My work is focused on the detection of the residual packaging toxins by using the fluorescence-based carbon quantum dots. Those are the sensors which used to the detection purpose, and they are synthesized by the use of the food waste. So we will detected the packaging residues by help of the carbon quantum dots. In present era, like the food safety is the major concern. So my work is all related to that. We utilize the food waste, or it aligns with the principle of the waste valorization that supports the circular economy also. So with that, this is not moving. Yeah, it's fine. So it's fine. Okay, so content which includes the introduction, review of literature, research gap, objectives, result and discussion, conclusion, publications and conferences. So first of all, I introduced about the packaging materials. Firstly, we use the packaging material in the food industries. They have the importance of the protection, preservation, convenience, information. They protect the food from the various contaminations or preserve the food for the longer shelf life, and the convenience for the storage or the storage purpose. And they provide the information to the consumers about the ingredients or the manufacturing dates or other composition of the food material. So the properties of the food packaging material, they have the barrier property, mechanical, thermal or chemical properties. As they have the barrier property which protects the food, and the chemical composition of the packaging material for the preparation of the packaging material, we use the plasticizers, stabilizers, slip agents, dyes. They help in the barrier properties also. But these all compositions like the plasticizer or stabilizers, there is a health risk from the packaging material to the human body. As the Ph.D. and EFSA, that is the Food Drug Administration, and EFSA, that regulates the safety standards about the packaging material, whether the food is safe for consume or not, or the packaging material is safe to use or not. So the emerging trends and innovations for the detection of the packaging material is the actives. We usually use the active or the smart packaging, like we use the oxygen scavengers in the smart packaging, or in the active packaging we monitor the food samples by the temperature or the color of the food material. So these are the emerging trends in the food packaging industry. After that, there is an interaction between the food material with the food packaging material. As during the polymerization reaction, during the synthesis of the packaging material, there are some of these packaging materials are formed by the number of these are the polymers and formed by the number of monomer units. As during the polymerization reaction, there are unreacted components there. So these unreacted components, they may migrate into the food materials, so that are harmful for the human consumption. And these are the phenomena through which these toxins migrate into the food products, that is the migration, absorption and the permeation. So these are some of the packaging components which are used in the packaging material, like the plasticizer, which increase the flexibility and durability of the packaging material. Then the stabilizers, they protect against the heat, UV light or the temperatures. And then the slip agents we add into the packaging material, which prevent the sticking on the metal surface. Then monomers and oligomers used in the packaging material for the barrier properties. Fillers we use to improve the visual properties of the packaging material. Then dyes which used as the printing inks for the consumer for the consumer information. So these all packaging materials, the examples are, some of the examples are like the bisphenol or phthalates we used as a plasticizer. They may affect the human health, like the ovarian disorders, like the reproductive disorders, or the immune disorders, cardiovascular disorders. So this is the mechanism of the cellular toxicity of the residual packaging components, like the toxins produced from the packaging material, they may react with the active site of the food material, active site of the cells of the human body. So some toxin, when react with the active site of the cells, they may get dead, or when the immune system responds or they generate the antigens against those toxins, then the cells remains live. Here we take the example of the bisphenol A, that is the stabilizer. When bisphenol A generates the oxidative stress, like the reactive oxygen species, they may damage the mitochondria or the DNA damage. So DNA damage, they induce the phosphorylation that result in the growth suppression of the cells. So then to overcome all these toxins, or to overcome these toxins which are migrate from the packaging material to the food, there are the number of the detection methods which evaluate or which detect those toxins. And some of the detection these techniques are the chromatography techniques, spectroscopy, colorimetric, biosensor. These are generally used, but the limitations of these techniques are, they are the time consuming, special manpower or the labor they required, expensive or advanced equipment they required. So we develop there is a need to develop some cost effective method. So we developed the carbon quantum dot by the help of the food waste material. So after that, what is the carbon quantum dots? These are the dots having the size below than the 10 nanometer. These are the nanomaterial kind of materials, and they are synthesized from the food waste, which are low in cost and used for the detection of the toxic residues present in the packaging materials. We can use these dots or sensors for other purpose also, like the heavy metal detections or pathogen detections also, but we detect the packaging residues. So the synthesis methods we used for the carbon quantum dots, we can use the top down or the bottom up methods. In top down method, what happens? Generally, the large molecules, they get convert into the smaller particles. In the bottom up, what happens? Those smaller molecules or the particles, they converted into the carbonized material. So we need the carbonized material for the synthesis of carbon dots. So we use the bottom up method, and in bottom up method we applied the hydrothermal method for the synthesis of carbon dots. Because the hydrothermal method, we need only the water and the heat treatment. We don't need any chemicals or anything, or this is the very simple or the cost effective method. So we use the hydrothermal method instead of the other methods. So the unique properties of the carbon quantum dot, they have the photoluminescence property. Like the photoluminescence means they have the ability to absorb the light or re-emit the light again. Then the biocompatibility and low toxicity means they have the ability to react with the bioactive biological molecules also with the less toxicity. Then the chemical stability, they have the chemical stability, they do not react with any other chemicals. Eco-friendly and low cost synthesis. And the quantum confinement effect, that is the quantum confinement effect is the confinement of the electrons. They change the optical or electronic properties of the molecules. So these all unique properties, they features the fast detection of the diversity, and they have the sensitivity, high sensitivity with the low limit of detections. Then the review of literature, I have reviewed the number of papers. Like for the we reviewed that the number of food waste materials utilized for the synthesis of the carbon quantum dots, but for the other applications like the in vivo electrochemical detection or the determining the antioxidant activity of carbon dots, H2 positive means a high metal contamination, we detect all these applications. So this is the review of literature of my work. Then the research gap of my research is, first is the detection of the residual toxins from the packaging material is still at initial stage. So, and the second is the detection of the residual toxin using the carbon quantum dot is still at an emerging technique, and the recommended dietary intake of the packaging component is still not developed by the FSSAIs. So these three are my research gap of my research. So with that, I'll start with my objectives. There are four objectives of my research. First is to synthesize the carbon quantum dots from the food industrial waste using the hydrothermal method. Second one is to characterization of those prepared carbon quantum dots. Third is to develop and validate with the analytical method for the detection of the toxic packaging components using the commercial standards. Commercial standards, what we used as a plasticizer for the detection purpose, and to detect the residual packaging components from the real food samples by employing the developed method. So these are the objectives of my research. Firstly, what we did, first we collected the food industrial waste samples like the onion peel, papaya peel, broken kidney bean, rice husk, walnut shell, liquid whey. Onion peel we obtained from the Punjab Agro Industry Corporation Limited, Chandigarh. Then papaya peel we collected it from the Punjab Agro Industry Corporation Limited, Chandigarh. Broken kidney bean we collected it from the Ajit Singh Om Prakash Limited, Nawa Shahar. Rice husk from the Mittal Agro Industry, Jalandhar. Then the walnut shells, we collected it from the Ajit Singh Om Prakash Limited, Nawa Shahar. Liquid whey, we collected it from the Verka milk plant, Jalandhar. So these are the all raw materials we used for the research work or for the synthesis of the carbon quantum dot. Firstly, we did the proximate analysis of all these samples to analyze the what is the content of the carbon, nitrogen or oxygen. That is like we have the number of the food waste material, there is a natural variability in the composition of the food materials, so there is a variability in the carbon content also. So we did the proximate analysis. And followed by that we did the EDS. EDS is for the elemental composition. So we detected that we estimate the carbon and the oxygen content of the different raw materials. So in the broken kidney beans, the carbon content is 58.08%. That is the highest one. No, not for onion peel, that is the carbon content is the highest one. So broken kidney bean, that is the 58%. Walnut shell, that is the 56%. And for the rice husk 31%, and for the onion peel 60%, and for the liquid whey, that is the 21%, that is the least one, and the papaya peel 49% carbon content is present there. So this is the method of synthesis of carbon quantum dots by the hydrothermal process. We use the hydrothermal reactor, that is a closed vessel, pressure vessel. We use that vessel for the synthesis of the hydrothermal. Firstly, we collected the food waste material, then the raw material powder we added it into the deionized water in the ratio of 1:80 weight by volume. Then we give the hydrothermal treatment at the 100, 150 and 200 degree degrees Celsius temperature for the variation with the time 246 in 10 hours, then we get the yellow suspension. After that, we did the filtration method or purification step for that yellow suspension. We use the, for the purification, we use the centrifugation at the 14,000 RPM for the 20 minutes. Then purification using the Amicon Ultra 15 3 kDa dialysis membrane centrifugal tube. Then we use the syringe filters of the 0.22 micron, and we got the carbon quantum dots. For the confirmation of those carbon dots, we did the visual confirmation under the UV transilluminator or by the visual confirmation or the UV spectrophotometer, particle size, and the fluorescence spectroscopy. And then these are the optimization process for the synthesis. We optimize the synthesis of carbon quantum dots at the different time and temperature intervals. So we optimized it at the 100, 150 and 200 for the 2, 4, 6, 8 and 10 hours. Then this is the visual confirmation of the synthesized. We obtained these samples at the different time and temperature intervals. So those selected ones are the dark in color, and those were not selected, those are in the light colors. So this is for the papaya peel derived carbon quantum dot. This is the visual confirmation of the kidney beans, and visual confirmation of the synthesized rice husk, visual confirmation of the walnut shell derived carbon quantum dots, and the visual confirmation of the liquid way derived carbon quantum dots. We selected the dark one samples for the further characterization. Then the visual confirmation of the synthesized under the UV using the transilluminator. Generally what happened, as you can see in the figures, we analyze the blue color of the carbon dots. This is due to the pi anti-pi bonding of the transitions. When we place a sample under the UV, what happened, there is a change in the transitions, like the pi bonding electrons go to the higher transition to the anti-pi bonding, then they re-emit to the ground state, and when they re-emit to the ground state, the fluorescence will fall down in the blue emission of the electromagnetic radiation. So they give the blue color under the UV. So we selected the dark color samples for the further characterization. So these are the images of the UV carbon quantum dots under the UV rays of the different time and temperature combinations. This is for the rice husk, this is for the walnut shell, and this is for the liquid way derived. So we selected only the dark samples for the further characterization. So this is the surface plasma resonance. This is we did this surface plasma resonance of the screened onion peel derived carbon dots under the UV visible spectrophotometer. So we get the peaks in the UV or visible regions only. So this is the LSPR of the UV visible spectroscopy of the onion peel. This is for the papaya peel carbon quantum dots. This is for the broken kidney quantum dots, and this is for the rice husk derived carbon quantum dots, and for the walnut shell derived carbon quantum dots, and the liquid way carbon quantum dots. Then the fluorescence spectra of the screened onion peel derived carbon quantum dot using the fluorescence spectroscopy. So here is the emission wavelength. What happened in the fluorescence spectroscopy? We excite the electrons at the absorption wavelength. When we excite those electrons at the absorption spectroscopy, they emit or they transfer to the higher wavelength of the frequency. That is the emission wavelength. So we get the emission wavelength of the 429, 526, 527. That is higher than the absorption range. So this is the fluorescence spectra of the onion peel derived carbon quantum dots, fluorescence spectra of the papaya peel derived carbon quantum dots, fluorescence spectra of the broken kidney beans, and the fluorescence spectra of the rice husk, and this is the fluorescence spectra of the walnut shell derived carbon quantum dots. So this is the fluorescence spectra of the liquid way carbon quantum dots. So then the particle size of the carbon, as I earlier told, that the particle size of the carbon quantum dot is in between the 1 to 10 nanometers. So at with the low time or temperature conditions, we got the particle size of the 16, 20, 10, but with the time or best optimized time or temperature condition, we got the smaller size of the carbon dots. As in the OPCQD 14 treatment, we got the 1.1 nanometer size. So this is the particle, this is the least particle size of the carbon quantum dots. Those are derived from the onion peels. Then these are the particle size of the carbon quantum dots, which are synthesized by the papaya peels, and this is the least one, that is 4.56. Then this is the particle size of the carbon quantum dots from the broken kidney beans, and these are the particle size images, graphs of the rice husk derived carbon quantum dots. These are the walnut shell derived carbon quantum dots, and the particle size of the liquid way carbon. This is the final table of the particle size, as we selected only the least particle size carbon quantum dots. So for the onion peel, we got the least particle size is 1.17, that is at the 150. When we synthesize the onion peel at the 150 degrees Celsius for the 10 hours, then we got the 1.117 nanometer particle size of the onion peel derived carbon quantum dot. For papaya peel, that is 4.156, and for the broken kidney beans, that is 1.22. Then the rice husk 6.06, walnut shells 2.08, and liquid weight 9.04. As the particle size is smaller, their properties of the fluorescence is good. So the least particle size have the good sensitivity towards the detection of the packaging material. So the objective 2 is the characterization of the synthesized carbon quantum dots. Those were the selected ones. So these are the selected ones, as we did the functional, to check the functional groups of the FTIs of the carbon quantum dots by using the FTIR spectroscopy. These are the some of the functional groups we detected, like the OH group, CC group, COH group. These are the functional groups we observed in the samples in the rice husk, walnut, liquid way, or other samples. Then we did the internal, to check the internal morphology of the carbon quantum, as the carbon quantum dots have the spherical structure. So you can see in the figures there are a spherical structure, but there is no agglomeration in the carbon quantum dot synthesis, so they have the higher stability. So then after that, apparent zeta potential of the carbon quantum dot, higher the zeta potential means there is a charge. To check the charge of the molecules, those were the molecules that are present in the carbon quantum dots, they have the particle, they have the zeta potential of the minus 11.6, minus 15.6, minus 12.9, minus 17.6. These are the apparent zeta potential of the carbon quantum dots. So then the thermal properties of the prepared carbon quantum dots using the DSC. DSC is the differential scanning calorimetry. So this technique is used to analyze the material, the resistance of the material, that how, at what temperature it will change their phase. So for the phase to check the phase transition of the carbon quantum dot, we check the thermal properties of the DSC. So at the 102 is the maximum, 103 or this is the maximum peak temperature at which they change their phase. So these are the thermal properties of the selected carbon quantum dots. Then the topographical image of the selected carbon. This is the topographical. This is analyzed by the help of the AFM technique. AFM is the atomic force microscopy. This tells us about there is a layers in the carbon quantum dot. So the height of the one layer is the 2.9 in the onion peel, 2.2 in the papaya peel. In the broken kidney bean, the height of the one layer is 2.6. Then the measurement of the carbon quantum yield of all the selected sample, we check the quantum yield of those samples by applying the formula or by applying the fluorescence intensity. So this is the formula, and we get the quantum yield of the selected samples. For the onion peel, the quantum yield was 46%, for the papaya peel that is 39, broken kidney bean 43, for the rice husk 24, walnut shell 17, liquid way. As you can see, the quantum yield is highly related to the carbon content of the materials. As we discussed in the EDS, we analyzed the elemental composition of these materials. There is an onion peel who have the higher carbon content, so we got the higher quantum yield of the carbon dots of the onion peel derived CQDs. After that, the objective 3 is to develop and validate the analytical method for the detection of the packaging component using the commercial standards. As we took these samples like the dimethyl phthalate, dimethyl phthalate, bisphenol A, epichlorohydrin, these are the plasticizers, stabilizers or fillers we add into the packaging material. So we selected all these packaging components for their detection in the targeted food matrix, that was the cookies or chips. So development of the detection method for the packaging residue using selected carbon dots as a fluorescence probe. Firstly, what we did there to check the fluorescence spectra of the onion peel derived carbon dots in the presence of the 10, we took the 10 micromolar of the bisphenol A, dimethyl phthalate, dimethyl phthalate and the epichlorohydrin. We dissolve it into the synthesized carbon dots, and then we check the fluorescence intensity and change in the wavelength. So this peak is the bare means without the addition of any residue. So this is the bare OP onion peel derived carbon quantum dots. When we add the 10 micromolar of the bisphenol or other residue, then they shift their wavelength towards the higher ratio. As the mechanism of the fluorescence is upon the quenching mechanism, there will be the quenching or there will be the enhancement in the wavelengths. So in our case, when we add any analyte or any packaging material, they will enhance their wavelength. So we can see the change in the wavelength by addition of these analytes. So this is the fluorescence spectra of the broken kidney bean with all the analytes. Then that is the fluorescence spectra of the rice husk carbon dots with the as you can see, there is a shift in the wavelength in the broken kidney bean, but in the case of the rice husk there is a no shift in the wavelength. As earlier we say that there is a quantum yield of those samples are higher, and the rice husk for liquid whey there is a quantum yield of less, or there is a carbon content, less carbon content. So they do not interact or they do not bind with the analytes. So they do not shift any wavelength or intensity. So they were not selected. So we selected only the onion peel, broken bean and the papaya peels for the detection purpose. Then to check the sensitivity of those selected carbon quantum dots towards the concentration of the packaging residue. Earlier we analyzed at the 10 micromolar, that was the highest concentration. But to check the sensitivity, we use the lower concentrations also. So we use the 0.1 molar, 2 molar, 0.2 micromolar, 0.3 micromolar, 0.4 micromolar and up to the 10 micromolar. We used all these concentration to check the sensitivity. So after the addition of these to check the sensitivity, firstly we influence the bisphenol A concentration upon the fluorescence intensity and the wavelength. So this is only for the bisphenol with the onion peel derived carbon quantum dots. This is for the onion peel with the dimethyl. This graph is for the dimethyl, and this is from the epichlorohydrin. So as you see, there is a no change, means up to 0.1, 2, 3 or 4, 0.4 micromolar we do not detect or we do not observe any detection because there is a no change in the wavelength. But up to the concentration of 0.5, 1, 2 higher concentration, there is a change in the wavelength or intensity. So after the range of the 0.5, we can detect this is the least limit of detection of the papaya peel carbon quantum dot towards the bisphenol A. So like that we check the sensitivity of the broken kidney beans with the different concentration of the packaging residues. Then the limit of detection, we get the limit of detection and limit of quantification by the linear graphs. We plot a linear graph with the higher correlation regression coefficient, that is the 0.9, and we got the limit of detection of 0.56461 for the onion peel carbon quantum dot, limit of detection for the papaya peel, that is 0.50484, and for broken kidney bean the least limit of detection, means the least concentration. What is the least concentration we can detect with our sensors? So these are the least concentration we can detect with our sensors. Then this is the linear graph of the onion peel, papaya peel and the broken kidney beans against the dimethyl, and this is against the epichlorohydrin. We can detect the limit of detection with the epichlorohydrin, that is 0.44418, 0.45, 0.56. Then the objective 4 is the collection of the different food samples from the local market and the optimization of the packaging residual components extraction method. That was the third objective, is about the sensitivity, to check the sensitivity of the developed sensor. But the last objective is the detection of those residue from the food matrix. So we selected the local food cookies or chips. So we detected the packaging inks which we spiked into the food materials. So for the extraction, first we spiked those analytes into the food material. Then after the food material, we extracted those packaging residues by applying the method Quetcher's technique. In the Quetcher techniques, what happened? We add the different chemicals like the magnesium sulfate, PSA, that is the primary secondary amine, and C18 into the food matrix. With the help of magnesium sulfate, that help in the magnesium sulfate that will remove the extra water, PSA that help in the removal of the polar matrix, or the C18 removes the non-polar matrix. So they help in the extraction of the packaging residue from the food material which we spiked earlier. So we optimized those samples also, those Quetcher's technique also. So for the optimization of the Quetcher's technique, we developed the different we take the different concentration of the PSA and the C18 and the constant concentration of the magnesium sulfate. So by adding all these components, we got the limit of detection, and for this, when we add the PSA of the 150 mg and C18 120 mg of the C18, we got the least detection, limit of detection. So we use this extraction technique for the further in our samples. So the extraction of the on the bisphenol A extraction from the cookie sample, we use this. Then for the extraction component on the diphthalate extraction from the cookies, we used also the PSA in the 150 mg or C18 in the quantity of the 120 mg. And then we optimize the extraction of the components of the dimethyl phthalates from the cookie sample. 150 or 150 mg of the PSA and 120 mg of the C18 got the best limit of detection, least limit of detection, that is the 0.63. and for the same in the case of the epichlorohydrin, 150 and 120 PSA and C18 we use for the least limit of detection. So in case of chips, we again we did the optimization of these Quetcher's techniques. We used the magnesium sulfate or the PSA or the C18. Again we optimized the best limit of detection for these components like the bisphenol, dimethyl and the epichlorohydrin for the extraction purpose from the chip sample. Then the extraction recovery of the packaging residue from the food matrix. This is the concentration which we spiked into the food sample. As we spiked the 10 micromolar of the bisphenol, 20 micromolar of the bisphenol and 30 micromolar of the bisphenol in the cookie sample. Then the recovery percentage is the 98%, 97% and 97.70%. Then the cookies, we added the dimethyl, same dimethyl and same dimethyl butyl epichlorohydrin into the cookie sample with the same concentration of the 10, 20 and 30 micromolars. Then in chip sample, we also spiked the different analytes like the bisphenol, dibutyl, dimethyl, epichlorohydrin at the different concentration, that is the 10, 20 and 30, and we got the recovery percentage of 98, 97, 96, 97 is the least recovery rate. So we detected all these packaging residue from the cookies and chips, and we validate that our developed sensors, developed method with the HPLC, and we recovered the 96, 95 and 97% after spike the 10, 20 or 30 micromolars of the packaging residue into the cookies and chip samples. So after that, after adding firstly, we did the characterization of the fluorescence particle size of the bare carbon quantum dots. Then to validate with after the addition of that there is any change in the fluorescence spectra after adding the packaging material, or any change in the particle size, or any change in the morphology, we checked under it in the fluorescence. So there is a change in the fluorescence intensity or the wavelength after adding those packaging residues. So these are the graphs of the packaging residue, and this is the visual confirmation. So we selected those dark one. After when we added the packaging residue, they get converted into the light colors, and this is the under the UV visible. And the particle size distribution, as you can check, when the carbon quantum dots, they have the particle size of the 10 nanometer, 1 to 10 nanometer, but when we added those packaging residues into the carbon quantum dots, the particle size will increase. So it will increase up to the 100 nanometer or 200 nanometers. So then we did the FTIR spectroscopy for the functional groups, as there are only the carboxylic groups, but when we add the packaging residue, there is a increase in the functional groups also. So we validate it with the FTIR, then the TEM. As you see, there are the spherical structures of the carbon dots, and as you see in these images, there is an agglomeration of those carbon dots. So this is the TEM images of the carbon quantum dots. Then this is the summary. I'll summarize this my research work. Firstly, with carbon quantum dots of the onion peel, papaya peel, broken kidney beans, rice husk, walnut shell, liquid whey, we synthesized at the different time and temperature combination, and based upon the visual evaluation under the UV light, UV spectra, fluorescence spectroscopy, particle size analysis of those carbon quantum dots were screened out. After that, onion peel carbon quantum dot, papaya peel carbon quantum dot, broken kidney carbon dot, rice husk carbon quantum dot, and walnut and liquid whey carbon dots at the 200 degrees Celsius for 10 hours showed the significantly lower particle size as compared to the other carbon dots. So FTIR confirmed the presence of the functional groups of the methyl or methylene amino groups, carbonyl groups that maintain the monodispersed nature of the carbon dots, and those selected carbon dots, they revealed the spherical structures during the TEM analysis, and they showed the surface charge in the range of the -11 to -22.2 during the zeta potential analysis. They showed the exothermic peaks and the phase transition in the range of 0.2 to 0.4 degrees Celsius during the DSC analysis for the thermal properties, and they revealed the topographical height and vertical dimensions in the range of 2.2 and 2.9 micrometer within a dimension of the 50 and 100. and 43, 24, 17, 12% respectively. Based upon their selected food samples, we selected the cookies and chips. For packaging toxic residues, we selected bisphenol A, dimethyl, dimethyl phthalate and epichlorohydrins were selected. After that, those selected onion peel derived carbon quantum dots, papaya peel and broken kidney bean carbon dot, they showed a shift in the intensity and wavelength in the presence of the selected packaging toxic residual components. And the onion peel carbon quantum dots, papaya peel and broken kidney beans, those were the selected. They showed a limit of detection and the limit of quantification value. The ratio of the cookies, magnesium, PSA and carbon C18 and the chips, magnesium, potassium, C18 showed the significantly lower limit of detection value for the bisphenol A, dimethyl, dimethyl and epichlorohydrin detection. And the ratio of the MPC4 that showed the significantly lower, where we use the magnesium is the constant, and PSA we use the 150 mg and C18 120 mg. They showed the significantly lower limit of detection value for the all packaging residues then from the food samples like the cookies and the chips. Then the recovery percentage of the packaging residue from the food matrix using the optimized HPLC method was observed in the range of 92 to 96%, and the recovery percentage of packaging residue from the food matrix using the developed fluorescence method was 97 to 99%. And after that we confirmed the visual confirmation with the confirmation of the shift of the dark brown to the light brown color in the presence of the standard and isolated packaging residues. Then the visual confirmation under the UV transilluminator that shift the dark blue color to the light color of the carbon dots after the presence of the standard packaging residues. Then we analyze the particle size analysis to confirmation the bigger size of the carbon quantum dots in the presence of the standard packaging residues. Then we did the FTIR and TAM, which shows us the different change in the functional groups or the aggregation in the carbon quantum dots in the presence of the isolated packaging toxic residues components. With that, we published the one research article in the International Journal of Biological Macromolecules that is related to my research work, which has impact factor of 7.7. Then the one more research article that is published in the Process Safety and Environmental Protection, impact factor having the 6.9. Then the one more research article in the Food Bioscience that is also related, that is for on the liquid way carbon quantum dots. And one review article that was published in the Chemosphere, impact factor having the 8.1. Then I did the international conferences in the Vignan University, in the Chaudhary Devilal University International Conference, and the one national conference in the Shulni University, and one national conference in the Himachal Pradesh University. Thank you.
I really appreciate the quantum and the quality of the work, really, and it's, mainly credit goes to your man, supervisor, and then is the student, because they have given the track.
Yes, ma'am.
And given the, you can say, acclimatized atmosphere,
yes,
under which you have conducted all these findings. No?
Yes, ma'am.
Okay, it's nice. Have a chair. We cover punch. चेयर बहुत आवाज करता है ना मैं चेक Okay, first you tell me the meaning of your name. What's the meaning of your name? I heard first time this name. Beautiful.
Actually मेरे father ने रखा था. She was an economist actually. Sweezee was an economist. About that, हाँ. My father did a MSc in economics. So
ये क्या हिंदी नेम है?
It's English. कोई फॉरेनर ही है कोई इकोनॉमिस्ट.
मीनिंग तो होगा.
हाँ मैंने खुद ही सर्च किया था तो that is sweet and sour. Sweet and sour ऐसे ही आता है.
It's good. Okay, can you tell me what type of structure of PhD is here in your program? Whether you have to clear the courses? Is there six months course? Just tell me something about your PhD structure.
Yes, ma'am. We did the coursework of 1.5 years. At our time, there was a 1.5 year coursework. In first year, they taught us the food subjects only.
We just name those subjects.
Food chemistry.
There will be some course number, then course title.
Yes, ma'am.
And what is the credit load for that? I want the complete answer for that. I'm not going to assess you for the thesis work. Yeah? Tell me the course number, course title, and the credit load. I think credit load is there, right? Yeah. It's theory, practical.
There was no practical actually. We did the coursework in the COVID. So there was that was online.
Okay.
Yes,
the course must be having some number and then course title.
Course title was food preservation and processing. Food chemistry was there, and nutraceuticals there, and the research methodology or the statistics, research writing, these programs were there.
These are the vague titles you are speaking or the exact titles?
Exact titles.
These are exact
Yes,
titles.
ma'am.
Not exact. But you must know.
Yeah.
You've already shared, na?
Yes, ma'am.
What was the total credit load for your these courses? You understand what is credit
Yeah.
load means?
Yes, ma'am.
It's the same?
Yes,
It's
ma'am.
3 plus 1 or 3 plus 2 or whatever?
At their batch, we were following the ICR pattern
ICR.
for 1.5.
That's nice.
Yes, ma'am.
कितना credit load निकाला आपने for your courses, then is for the research?
Their credit.
Yes, ma'am.
यही पूछना हमने बस then free.
अब पता चला है क्रेडिट लोड, उस टाइम तो पता ही नहीं होता था क्या होता है
You must
क्रेडिट
know
लोड।
it,
Yeah,
and then you will be
obviously we
able
must
to
know.
again now
Yes.
you are going to be ready from this side of the table to this side, na? So you must prepare yourself for that. Okay. So again it is major subject, minor subject. No, it's a major one.
No.
For food technology, we were in their batch, we were following the ICR system like in Ph.D.
In Ph.D.
So C was there.
Okay. Can you tell me something? Strength. Have you ever done your SWOT analysis of your own?
Yes, ma'am.
Your strengths, your weaknesses,
Okay.
which have enhanced your during your study period, or the weaknesses which you got the solution to overcome those?
Yes, ma'am. In PhD, as we should be patient, so I was very patient at that time, and obviously my sir, Dr. Prince Chawla, he supported me in my down time, or है ना, तो that was the main reason कि I was very patient, or that was the
That was your
strength.
strength.
Yeah, that was my strength.
Any weakness? Weakness तो होती है सभी में.
Yes.
तो क्या है आपकी?
I think I can't express, मतलब जितना मुझे आता है, I can't express that easily.
So now that you got the solution, how to overcome that? Because
Yeah,
okay.
very good.
Okay. Just about your this, you said this carbon dioxide act eco-friendly.
Yes,
How
ma'am.
you justify that? Carbon?
Because they are the food waste, so we use the food that is the eco-friendly in nature. There is no chemical composition in that. They are naturally available. So, and we use only the water in the synthesis process, and we give the heat treatment.
How you define eco-friendly?
Eco-friendly
I
means
think
that
second or third
Yes, ma'am.
you mentioned that.
Yes, ma'am.
In your introduction.
That will not affect our environment. So that is the.
Okay. So I have just you have collected these your different food waste from different places.
Yes, ma'am.
So how much time you took for that? Their generation of the waste and your collection, and then your pre-processing here itself in the laboratory. How much time you took that?
Yes, ma'am. It take only
Because
the one. Yes,
in all your, I think there were 6 your waste.
Yes, ma'am.
Of different nature.
for the
right?
synthesis of carbon dots, we took
No, I'm
the
not saying
only
about
the for the.
I'm not saying about the carbon dioxide.
Yes, ma'am.
You went to those industry
Yes,
or
ma'am.
places.
Yes, ma'am.
So कब generate हुआ था, कब आपने collect किया, and you took it to your lab and you did the pre-processing for that.
It take around 1 week. We go to that industry, then we collected that, and after second day we take it for the in the hot air oven for the drying purpose. Yes, ma'am. After that we did the carbonization or the carbon content. We took the carbon
So
content.
it's same for all type of-
The samples. Yes, ma'am.
Okay, so which was the period when you just collected these raw materials? What was the temperature, humidity?
Yes,
What
ma'am.
were the conditions over there?
Yes, ma'am. That was.
And in which packaging material you have collected those
Yes,
waste?
In the March month, we collected those samples in the Ziplocs, plastic
Ziplocs.
materials. Yes, ma'am.
Have you assessed their initial any toxicity or any microbial load of those waste?
No,
Have you done
ma'am.
such type of analysis? You've done the proximate composition,
Yes,
that's
ma'am,
after
we did
drying. But immediately after the collection, have you done any microbial analysis from the
didn't check in.
safety point of view?
No, we
What I
don't
felt, I think you
but-
could have done it, right? You did the drying next day, ना?
Yes, ma'am.
वो किस कंडीशन में आए, कितना उनका इनिशियल मॉइस्चर था,
Moistures,
whether they were safe or not? ये थोड़ा सा चेक करो, it's fine.
बीच में कोई स्पॉइल्ड वाला था या नहीं कि ये सारी चीजें।
Okay, on the whole, from your whole study, can you tell me what are the strength and limitation of your study?
Yes, ma'am. Actually, we worked on the waste valorization, है ना? So we worked on that. We utilized the waste material and convert into the value-added materials, to the nano materials. So we utilize the waste and we prevent the pollution and utilized in the.
And any specific limitation of your study?
Yes, ma'am. We checked actually the stability of these carbon quantum dots. That will degrade after some time, so we can use that in the initial times.
Any hurdle which you faced during your?
Yes, ma'am. On the particle size, we face the hurdles. Starting at the initial time, we got the maximum that particle size. After, with the optimization, we check the particle size, but we change the time or temperature conditions. With that, we get the lower particle size.
So you have done many of your detection method, new detection method. What do you think these current detection method for any packaging toxins, they are better than the previous studies or previous methods?
Yes, ma'am. We developed these for the cost-effective method only. So generally, HPLC or GCMS, they are also used for the detection purpose, but they are the highly labor, I think, or very expensive. They are in cost. So we synthesize these carbon dots from the waste materials. So this is the method, actually a method developed. We developed a method which we can apply for the fluorescence for the detection purpose. This is not a method. Yes.
You are saying this HPLC, they are the costlier method.
Yes, ma'am.
And your method, so कितना cost cutting हुआ? What we can suggest that?
For the HPLC method, there is a cost for the instrumentation. Like we can take the example of the CIF of LPU. When we analyze the sample, we send the sample for the HPLC, there is a cost of 1500 or 2000 for the HPLC. And when we analyze the any sample under the fluorescence, there is a cost of 50 rupees only. So
What
that
do you
is
think
the
this
major.
industry, any packaging industry, they will be able to, can you convince the packaging industry to go for this component rather than going for this
Yes, ma'am.
advanced techniques?
Actually, we check the sensitivity. That is the major issue. Sensitivity, we got the good sensitivity of our compounds in relative to the other ones. And one is the cost. Cost is the main, so we can convince on those factors.
But I think still they must be go for the validation of the results. Again with the advanced or the newer methods.
Yes, but in that we also validate the HPLC.
Yes, you will. अच्छा था मैंने I have gone through with your thesis मतलब even I learned a lot from this component and even आज तो और कई concepts clear हो गए कितने questions मेरे mind में थे तो I think I have got the answer for that. You have very well justified the reasoning components. That's I really appreciate the way advisor has guided, your guide has guided to compile and justify the results. Okay. Can you tell me, let's say your study, your findings, they will be on float, right? How this packaging industry will be benefited from these findings or recommendations?
Yes, ma'am. Actually, we did the detection from the local foods only because there are many chances of migration of those inks, local inks maybe. You know, they can migrate into the foods. But in the packaging industry, we can talk about the packaging industry. There is a less chances of migration. But after after the expiration date or the we can detect if there is some lot or something when they do the polymerization reaction, they form those packaging materials. Sometimes there are a chances that unreacted components may be there, so they will migrate into the food materials, where there we can apply these
Here in the
methods.
pics you have shown this packaging material of your chips and of the cookies.
Yes, ma'am.
What was the packaging material in case of your cookies and as well as in case of your chips?
That was the plastic.
Plastic.
LDP.
LDP.
Yes,
क्या होता है LDP?
Low density polyethylene.
Polyethylene.
Yes, ma'am.
So why you have not taken any packaging material which is available from the say आपके जो branded companies के जो biscuits or chips they are available?
Like laminated.
Laminated one or the.
Yes, ma'am. There is a less chances of migration.
How we can
but-
say it? What do you think? And do this FSSAI they are having any specifications for these?
No, ma'am. There
was a gap in your.
Yes, ma'am.
there was
That is a gap. There was one newsletter or one news cutting. I showed in the first slide also that the FDA required some techniques or methods for the detection of these hazardous materials, for the detection of the hazardous materials. This is the major concern. That is the cutting of last 2020 only.
So you have done like very quantum and quality really appreciated. Everything is done, right? You have published, you have presented at different platforms. Initially you mentioned 3 gaps.
Yes, ma'am.
Right? What were those gaps? Can you name 1, 2, 3?
Yes, ma'am. That like the recommended daily intake, the detection of the packaging residue is still at the emerging stage, or the detection of the packaging residue by applying the carbon quantum dot is the emerging stage, and the FSSAI. Yes, ma'am.
Okay. 3 gaps initially by planning or executing this whole study. So and now at this stage, after publications, after your presentation, what do you think, to what extent we have achieved the success in filling those gaps?
Yes,
Which
ma'am.
will be beneficial to the scientist, even they will get the direction, or it will be for the food industry or the packaging industry,
Yes, ma'am.
stakeholders.
Earlier, as I reviewed the number of papers, so there is no detection of packaging residue by applying the carbon
that's
dots.
fine.
Yes, ma'am.
Not now, but in later stage where you must assess it. Okay, these were the gaps. We
Yes,
have
ma'am.
done this component. Now, to what extent we have achieved to just fill that particular gap?
Particular gaps.
Or how we can extend this study?
Yes, ma'am. We can use like packaging. We can improve the quality of the packaging materials. Firstly, improve the packaging material synthesis or the polymerization, those reactions, and after that we can use those carbon further more sensitivity, as I detected up to the 0.5 molar micromolar quantity limit of detection. The least limit of detection was that 0.5 micromolar, but we can check the more sensitive, we can prepare the more sensitive materials also that can check the 0.1.
नहीं ये शो नहीं होते ना.
अच्छा इनको
यादी के पास
अच्छा चलो I think the expert they have mentioned some questions. It's one is very good, that how did you ensure the reproducibility, and number 2 it is the accuracy of your experimental data.
Accuracy, precision.
You've done the validation?
Yes, ma'am. I done the validation. We checked the fluorescence intensity. By those fluorescence intensity, we plotted graphs in the Excels, and from there we got the linear graphs. From that correlation regressions, we checked the accuracy, and we detected that the limit of detection and the limit of quantification. Limit of detection is to the least concentration we can detect it, and for that limit of quantification is for the accuracy and the precision purpose, that how much are these dots are accurate, or the how much is the precision for that. For that we check the limit of quantification.
Okay. So you have your findings, you have your recommendations. What do you think, what type of challenges which you face to take away these findings to the end users or to the stakeholders or to the industry persons? Or it will be limited only to the publications or this level?
Oh, ma'am, definitely. I want to.
Will you be?
In your opinion.
Yes, ma'am.
And any two major challenges which you may face?
First is the synthesis. First was the synthesis, and second was the sensitivity.
Now you have all the method. Ma'am is saying that how you are going to take it to the end user or the stakeholder who are going to actually use this technology or method.
इसको आगे ले जाने में क्या you think challenges आ सकते हैं?
Maybe हो सकते
Smooth हो सकती है. या smooth से नहीं होगी और
Smooth.
I have said to you say no, then justify.
कोई challenge नहीं है.
कोई challenge नहीं है.
ये mind की this challenge. जहाँ मेरी रिफाइनमेंट ऑफ टेक्नोलॉजी की रिक्वायरमेंट ये मैंने कुछ इसको अब देखो कितने आपको हिंट दे दी.
Yes, ma'am. There will be no.
No.
I think in recent scenario we should focus on just strip-based methods or just quick methods. So this is not one of the quick methods.
Yeah.
Like fluorescence equipment is desired over here. Yes, something like visual confirmation can be done. So next can be done like that. We can use carbon quantum dots and with our eyes only we can detect them.
This is how you will take this study in the future.
Future searchable issue.
And second thing is FSSA approach.
Yeah.
That is very limited towards us. Like we have to approach them that yes, we already designed or discovered one method or developed one method, so that it can be commercialized at a large scale.
Exactly. Do you say that?
LDP की जगह भी कुछ कहा था.
Laminates.
आपने use किया.
Different packaging materials.
Yes, ma'am. Laminates.
वो भी
That component can be.
Yes, ma'am. Yes, ma'am.
उसी में आ गई है next.
Okay. एक आपका था कि वो जैसे मैंने बोला था कि इनिशियल माइक्रोबियल ग्रोथ जो आपका रॉ मटेरियल था, you could have done that component. Okay. I think I have seen in all your figures and in tables you mentioned the sample code, but I think we could have given the description, isn't it? This runs over here.
नहीं वो इन्होंने ग्राफ्स के नीचे तो वो
नहीं, it's again a code OP and all.
Onion peel.
अच्छा, onion चलता है आपका यहाँ पे.
It is mentioned.
And even in जैसे table
Abbreviation use.
Abbreviation use किया
figure
और
2021 में भी आपने लिखा ना, you have given the sample codes ना.
Yes.
But in my words, I think that your tables and your figures should be self-explanatory, ना?
Okay.
मतलब I should not go for मतलब जो भी पढ़े उसको देखने के लिए पेपर पब्लिकेशन में कि हमें ढूंढना पड़े OP means this, okay, B2, B3 is this, ना? थोड़ा ये देखेंगे. ये components, good. And एक के अपनी जैसे figure 39 और 40 में देखा था, you used term this packaging residue.
Yes, ma'am.
How you define it, packaging residue?
In packaging residue, we can take the plasticizers, stabilizers, so we optimize for the bisphenol, that is the plasticizer.
Will the term will be packaging residues?
Residue means, ma'am, the small micro particles of that packaging materials. Like she already explained that there are free foam. Unreacted means free foam of the packaging material. They are actually migrating into the food products. So they are not the larger one size. Actually, we are eating along with that. So that are in very small size. Micro size which are not visible.
Whenever you got this term, na, residue, that's a bit, it looks like a bit alarming. Na? That's why just in titles you mentioned packaging residue.
Yes.
If you can use some modified term for that. कि थोड़ा negativity की तरफ वो Okay, and then is the one is in table 15 में था आपका जो retention of packaging residue. Okay. Explain that component. So but that particular table आपका 15 में था, how much has been retained in that? Retention of your packaging residues का था. Can you tell me कि if you want to convey to some consumers in layman's word, how you will be able to convey that? Okay, this is safe up to this limit, it's okay, and beyond that it will not be.
But in that we use the 10 micromolar, that is the higher concentration we use first to check the sensitivity. And for the sensitivity we use the lower limits also, like the 0.1 micromolar, 0.2 micromolars. But this is the method we developed for the to check the sensitivity of the packet.
तुम अपनी
But
स्टडी में
how
जा रहे हो,
to-
मैम का क्वेश्चन बड़ा जर्नल
हाँ, कि
है,
उनको-
हाउ टू कन्विंस द लेमैन
Layman.
कि जो आप रेजिडुअल की बात कर रहे हो
हाँ
या
जी।
रिटेंशन की बात कर रहे हो, आप कैसे उसको कन्विंस करोगे कि जितना है इसमें वो सेफ है?
Toxicological studies are required.
Required.
Most studies-
And same way
We developed the only
जैसे हम
sensors.
आजकल बोलते हैं ना प्लास्टिक बॉटल्स आती है, वाटर बॉटल्स आती है, नॉट generally it says that you can't keep it much time for
Yes,
living.
ma'am.
Maybe in the
Maybe in
refrigeration.
that also
What
residues,
packaging-
unreacted
What's
residues
that
are
packaging
there.
material? The
Which
water bottles?
For water bottle,
हम बोलते हैं ना PET bottles.
yes, ma'am,
What's
yes,
that
ma'am,
PET means?
polyethylene.
And they used to say that this water, it doesn't, मतलब it should not be at 35-40 degree में.
Yes,
Continuously it
ma'am.
should
They
not be
mild.
2 months.
They have the thermal properties.
What happens?
They
क्या
are
होता है?
resistant to that temperature
Some
only.
toxicity develops. क्या
Yes,
किस
ma'am.
कौन सा कंपाउंड डिग्रेड हो जाता है उसमें से?
वो भी बेसिकली हम माइग्रेशन की
माइग्रेशन की बात है ना, तो आपका ये कार्बन
हाँ
डॉट्स
जी।
दे कैन बी,
Yes, ma'am.
मतलब आप इसको
For-
भी
they can
आप ना यू कैन
use to detect those-
डिफरेंट पैकेजिंग
those
मटेरियल्स में
residues.
आप लोग ट्राई करके
Yes,
इसको कर
ma'am.
सकते हो further.
प्रेजेंटली दैट्स द मोस्ट इम्पोर्टेंट नाउअडेज.
Yes, ma'am.
देखा ना आपने, जैसे when we are traveling on the roads, वाटर बॉटल्स की बॉटल्स आपकी वो पड़ी है, मतलब ना that's
Yes.
under, they are not under shade,
Yes, ma'am.
not in the refrigerator, so that is dangerous component. That's- but I think if it's possible also you can extend your study
Exactly.
to your master students
Sure.
for these water bottles component. Anything, the beauty of your study or beauty of your whole journey of your PhD?
It's like I develop carbon dots from the food waste. So waste well, yes, ma'am.
Total during
Waste.
PhD,
Yes, ma'am.
special salient
कोई कुछ बताइए
आपको
हमें.
अच्छे पॉइंट्स जो फील किए.
That's will remain in
Study के
your
अलावा कुछ नहीं।
study के बाद तो
जनरल क्वेश्चन को
स्टडी से निकल नहीं पा रहे हो तुम, स्टडी को छोड़ दो, इन जनरल
Yes, ma'am.
बातें हो रही हैं अब तो.
फ्री कोरियर का जो टोटल स्टे
हाँ.
आया टिल वो सब from your joining
We learned a lot from Milpio or
सभी करते
related.
हैं. अब यहाँ बैठे हमें अच्छा कहना ही पड़ेगा.
3-4 साल लगा दिए तो इसने सीखा ही है.
स्पेशल
Anything beauty of your component?
मैं एक चीज कहूँ इसमें, सबसे न्यू एरिया, सबसे अच्छी ब्यूटी ये है कि तुम प्रोडक्ट डेवलपमेंट में ना जाके तुमने एक बड़ा नया एरिया जो है ना उसको लिया और उस पर काम किया.
And that too during PhD.
Right.
This is characterization. It's the most important. Generally we go for this basic or applied component, ना? So that's really.
मतलब हमें पता है कि तुम्हारी स्टडी की ब्यूटी क्या है.
हमने बहुत लर्न किया बेटा.
That is mainly interdisciplinary or something like that. no?
Because this is basically when we say FSSAI का हम कह रहे हैं, this work is basically for regulatory bodies.
Yes, ma'am. Regulatory.
They can use that and they can you can say publish your method for the detection purposes.
ये क्या है? What is FSSAI? मैम ने बोला,
Yes,
हमने
ma'am.
बोला,
Food
आपने बोला.
Safety Standard Authority of India,
क्या? When it has been established?
2006.
6.
Yes.
What is, how you define food technology?
Food technology refers to the which technologies we used for the food processing.
Exactly, मेरे को लगता है मैम ने definition जरूर आपको लोगों को specific definition for food technology. How you define food technology?
The technologies we used for the food processing or to preserve the food for in the food packaging also. No?
Now?
वो ये जो देता है ना साइंटिस्ट का नाम देता है.
हाँ हाँ.
Who has given this definition?
Complete definition.
It's a complete discipline.
तो आप इसे बोल सकते स्पोर्ट के लिए.
Okay. Can you tell me any book which you purchased during your graduation, your masters and your PhD? कौन सा book purchase किया? Have you purchased those?
Yes, ma'am.
Yeah,
Yes,
बताइए.
ma'am. Whitaker
Whitaker.
हाँ जी.
Whitaker की book किस
Whitaker की that is for malting and brewing.
Okay.
हाँ जी. And fermentation के लिए Kent की book and principles. Wheat, sorry, cereal. Kent is for cereals.
Okay,
Malting and
I'm
brewing.
going to
Whitaker is for fermentation. Sorry. And the food preservation principles of food Shakuntala मैंने
Oh, उसका नाम
सुकुमार dairy technology.
चलो just tell me one book which you will say to your undergraduate students, to your master students and to your doctorates. one book each.
नेट एग्जाम क्लियर करना है तो कौन कौन सी बुक्स पढ़नी है?
Sukumari is good for dairy
वो
technology.
तो चला गया यूजी लेवल का हो गया, आगे बोलिए. For master's level and for the doctoral level.
Yes, ma'am.
If you are to recommend the book to your students.
PhD में अभी advanced
Advanced food chemistry and
फूड केमिस्ट्री में कौन सी बुक रिकमेंड करोगे?
food chemistry में authors. Yes, ma'am.
और ये तुम लोग सारे तैयारी कर लो।
Nutraceutical.
ये तुम्हारे लिए क्वेश्चंस है, तैयारी करो, पता होना चाहिए अपने
So how you've gone for I think 4-5 publications. How you have selected these journals? Okay, this journal should be there. What's basic the criteria which you require for that?
We check that is related to our research work
वो
on
तो
food
general
safety.
हो गया.
Food
और उसके
safety.
अलावा क्या देखते हैं हम? N number of journals are there in food safety. Still you select the. क्या
It is
what's the
related to
criteria?
the food also.
हो गया.
Yes, ma'am.
आगे safety भी हो गया.
आपने गाइड ने बताया सीधे ही तो आपके पास आंसर।
तो फिर आपने सर से पूछा नहीं कि why this journal, why not this one? क्या criteria होता है जिस basis पे we can select the journals? Let's this be the common question for all. Scopus
और
और
LQ डिस्कस
यहाँ पे
ये रिक्वायरमेंट
यहाँ पे
Yes, ma'am.
और स्कोपस के लिए भी वो काफी वेरिड है ना, तो क्या देखते हैं हम लोग? Impact factor. Okay. How you define impact factor? How this impact factor is being calculated?
The quality of the journal.
नहीं कैसे कैलकुलेट करते हैं? अभी इसका इम्पैक्ट फैक्टर 3.6 आपने बताया, 6 था, 6 था, किसका 7 था, कैसे कैलकुलेट होता है? साइट स्कोर वो कैसे कैलकुलेट करते हैं? नंबर ऑफ
हाँ, scientists.
पर चलो आई थिंक दिस इज ऑल फ्रॉम माय साइड. यू कैन कंटिन्यू आल्सो प्लीज. करो प्लीज.
आपके साथ ही पूछ लिया चल रही हो हाउ यू रेट योर evaluation part self rating
यस
I should improve my
नहीं
expressing or rating.
out of 10 8 9 10
It's 10.
10 out of 10
Yes. No. Yes, ma'am. We will give.
10 out of 10 तो मेरा नहीं जस्टिफाई
नहीं जस्टिफाई.
Ne-
ठीक है
Yes, ma'am.
तो
तो ओवर कॉन्फिडेंस हो गया भैया.
जेन्युइन रेटिंग कैसी है
8.
8
Yes, sir.
एक आपने proximate composition में why we wrote it proximate composition term क्यों use करते हैं? Let this be the common question for all PhD scholars. Why we name it as the proximate composition? And second question is you wrote it crude fat.
Yes, ma'am.
Then it is a protein. Why this protein you didn't wrote it as a crude protein? नहीं लिखा आपने, प्रोटीन लिखा क्या? वो क्रूड हो गया, ये क्रूड ना हुआ. क्रूड मीन्स
मैम उससे हमने रिमूव कर दिया, लाइक फैट को रिमूव करते हैं हम पहले ना, प्रोटीन.
there are two terms na, crude protein and true protein.
Yes, ma'am.
So you have estimated crude protein or true protein? Is it a crude protein or true protein?
Crude protein.
Crude protein.
Yes.
How you define crude protein?
एक्सट्रैक्टेड बननी है वो, मतलब प्रोटीन जो आ गया दैट वाज द एक्सट्रैक्टेड फ्रॉम द एंसेम्बल्स, और क्रूड इज द नेचुरली दैट इज प्रेजेंट.
Which method you use for that?
फॉर प्रोटीन एस्टिमेशन यस मैम,
Okay.
जेल डाल.
Gel dal.
यस मैम.
जनरल में क्या करते हैं? डायरेक्टली प्रोटीन कुछ और पहले कैलकुलेट करते हैं?
नाइट्रोजन. नाइट्रोजन
Actually form of that protein, it does not contain any residue of other components.
नहीं, crude means rough.
Crude means
उसमें
rough.
सब कुछ आता है, कहीं इधर से कहीं इधर से. नाइट्रोजन आपकी आ गया,
नाइट्रोजन.
नाइट्रोजन कहीं से भी आ गया.
नॉन नाइट्रोजन ये नॉन प्रोटीनेशियस सोर्सेज भी आ गए ना, तो इसलिए क्रूड है वो.
मैम ने तो कह दिया नाइट्रोजन कैलकुलेट करते हैं, इसने प्रोटीन कर दिया. तो फिर अगर कैलकुलेशन आपका नीचे नाइट्रोजन आया तो प्रोटीन कैसे निकल आया नाइट्रोजन से? कहाँ से आ गया प्रोटीन के?
उसका फार्मूला
फैक्टर नहीं लगाते, 6.25 लगाया होगा, 5.92 लगाया।
नाइट्रोजन नहीं होता स्ट्रक्चर तो अमीन ग्रुप प्रोटीन में लगा होता है, वो एवरेज है 6.25, यानी कि हम एवरेज मान रहे हैं आउट ऑफ 100।
16% प्रोटीन लेके चल रहे होते हैं हम।
13 से 19 है सब ग्रुप मटेरियल्स में। तो अब जो है अकॉर्डिंगली आफ्टर 1940 जो है वो चेंज हो गया फैक्टर।
हाँ,
अब वो 5.39 से 6.38 है। सब मटेरियल्स का अलग अलग करेक्शन फैक्टर है, वे के लिए अलग जाएगा, मिल्क के लिए अलग जाएगा, सब के लिए अलग अलग जाएगा।
When you entered this room, what was running in your mind? मतलब what you were expecting? और वो हमने ना पूछा हो, हमें पता ना हो क्या चाहते थे आप, कोई ऐसा क्वेश्चन है कुछ?
बिफोर एंड आफ्टर। बिफोर बताओ। बोलो
Sir,
साइन ही करें मैडम, कान से सुन रहे।
when I entered, I thought it will be good. Now, yes, ma'am. I was comfortable.
It's good. God bless you.
Thank you.
What's plan? She has joined a meeting, ma'am. A meeting in Mohali.
Mohali.
वेरी गुड, कंग्रेचुलेशंस।
Thank you.
अभी क्या?
In September month. After pre-submission, I joined in September 24th
As in?
as an assistant professor in FSP.
अभी जो उनकी
हाँ,
पोस्ट
अभी
अपनी
September।
कंट्रेक्चुअल में किया कि परमानेंट में?
हाँ जी, contractual.
कंट्रेक्चुअल।
वेरी गुड।
वेरी नाइस। अभी उनका परमानेंट में कल इंटरव्यू हुआ ना उनका?
अब इसके पीएडी के बाद
अभी था। इनका
अभी हुआ है।
दिसंबर में हुई इनकी।
हाँ जी, हो गया वहाँ पे फूड इंजीनियरिंग के लिए अपॉइंट
फूड
हुए
इंजीनियरिंग के थे।
हाँ जी,
2 पोस्ट।
हाँ जी, अभी अपॉइंट हो गया,
Appoint
एक्सेल
हो
आ
गया?
गया।
आ
हाँ
गया?
जी, आ गया।
उसमें थी एक्सपर्ट।
मैंने
अच्छा, तभी मैंने कहा कि वो हाँ,
मेरा सितंबर
मैडम इसका ध्यान रखना नेक्स्ट टाइम जब आप जाएं।
बहुत अच्छा था वो लोग। सो उसमें ना जो कैंडिडेट्स थे ना, दे वर सो गुड। आई थिंक दैट इंटरव्यू का जो पूरा सेशन था, इट रन फॉर 6 to 7 hours। बहुत अच्छा है। ऑनलाइन चलो इसका हमें आंसर बताइए। हमें तो आता ही नहीं क्या लिखना। द कैंडिडेट इज रिकमेंडेड फॉर द अवार्ड ऑफ द डिग्री। द वाइवा वॉस एग्जामिनेशन इज नॉट सेटिस्फैक्टरी। द कैंडिडेट इज advised to appear in वाइवा एनी टाइम विदिन दिस। क्या आंसर होगा बेटा इसका? व्हाट शुड बी द आंसर? कट कर दूं इसको? हाँ, इसको कट कर दूं? इट्स कर दी। हम नीचे कर दी।
क्रॉस कर, क्रॉस कर,
हम इनके क्रॉस करते हैं।
क्रॉस ही कर, एग्जैक्ट क्रॉस कर।
स्टेटमेंट्स में कैसा कि कुछ
मैडम
स्टेटमेंट्स
फ्री कर। मैडम इसको फ्री कर।
हाँ जी
नहीं सर, अनाउंस करवा देंगे मैम तो।
अनाउंसमेंट डॉक्टर साहब करेंगे प्लीज।
मैडम जी साइन हो गया ना, टाइम हो गया इसका कि मैं
So now the candidate is recommended for the award of the PhD degree.
Congratulations.
Credit goes to your mentors, your teachers, and the advisors. Really, congratulations. And continue hard work.
Yes, ma'am.
And try to concentrate on the basic component much more. So you must be clear with everything. Now you're going to be on this side of the table. Right? Take care. God bless you all. God bless you.